All right, well, thank you everyone for joining us on our latest edition of Wildlife Wednesday with the Center for Wildlife Studies. This time I'm tuning in from near Seattle, as I just said, a little escape from Alaska for a while. This program is intended to, we invite researchers and educators to come present about their work to us and just share what's going on and help recharge our batteries. Is one of my big motivators for this during this pretty challenging time. My name is Jeff Stetz. I'm the Director of Science and an Assistant Professor at the Center for Wildlife Studies. I'd like to thank everyone again for joining us. This is obviously a tough time for some folks, but it's spanning pretty much around the globe right now. Just to remind folks, if you like, we do record these and post them to our YouTube channel. I'll put a link in the chat for that. So if people aren't able to join us live, please share that link with other folks. And we're building a little library there of our presentations. While there, you could also check out our course offerings and our professional certificate programs. We're trying to grow both of those programs pretty quickly right now. And we're also launching a science services branch to CWS, where we'll be trying to assist folks with a range of skills, whether it's conservation planning, study design, publishing results, statistical analysis, whatever we can do. We're hoping to build that program over the coming years as well. But no one's here to listen to me talk, so I'd like to introduce Dr. Natalie Hamilton. She'll be talking to us today about her research leveraging natural history collections to investigate a broad range of applied ecological questions. Natalie received her bachelor's in biology in 2015 and her master's in evolutionary and ecology and organismal biology in 2019, both from Ohio State University. And she was awarded her PhD in wildlife and fishery sciences from Texas A&M. And that was 2023, I believe. Currently, she's a postdoctoral researcher at the University of Alaska Museum in the mammalogy department with Dr. Link Olson. And how this came to be is I tried to reach out to Link a few years ago to talk about some research ideas in Alaska and how we could put their amazing collections to work for the state in developing some new research projects and so on. And when I reached out to him to maybe present to our program here, because I think some of that work is just fascinating and very much underappreciated, he immediately recommended Natalie, which I thought was very high praise coming from Link. But as we're here to learn from Dr. Hamilton, I will hand it over to her. I will again note that these presentations are recorded, and I'll also put a link to Natalie's research site if folks want to look into more of her work, which what I've seen has got some pretty cool stuff, including research on bats and population genetic structure of marmots across North America. Some pretty interesting work for sure. So please check that out. And with that, I think we're all geared up for Natalie to take over. So I will mute and keep watching the waiting room. Take it away.
Okay. Thank you for that introduction, Jeff. Yes, as you said, I'm a postdoctoral researcher at the University of Alaska, and I'm just going to talk about some research that I've done during my PhD and currently with collaborators at Texas A&M. So I like to start out my talks with just sort of talking about my collaborators, just in case I run out of time. Science isn't done by one person, and these are the PIs that are involved with this project. Dr. Jessica Light and Dr. Sharon Janza have, helped us a lot through this project. So I just wanted to, recognize them up front. And then I did a lot of the genomic research for this project, but Brandy, Haley, and Grace are doing all of the morphological research. So I just want to, give them a shout out before we go. And then there's just been so many people involved in this project. There's a lot of mice, there's a lot to be done. So I just wanted to thank them in advance. And our study is focusing on mice in the genus Peromyscus. So these are commonly known as deer mice, white-footed mice. There are over 50 named species in North America, some of which I'm showing here. So each of these named clades corresponds to a species group. And our study focuses on the Peromyscus maniculatus species group, which I've shown here. There are 9 species within this group. Here they are. You'll hear me talk about them, but just wanted to show them: Arcticus, Gambeli, Kenai, Lobecula, maniculatus, melanotus, Polinotus, Sejujus, and Sonoriensis. So my talk kind of focuses on those 9 species and the species group and their evolution. So the North American deer mouse, Peromyscus maniculatus, is one of the most widespread rodent species in North America. So it has a range that extends across most of the United States and Mexico, and large areas of Canada. And it was once noticed that within the range of one species, it's probable that a line or several lines can be drawn from Labrador to Alaska and then to southern Mexico, throughout which not a single square mile is not inhabited by some form of this species. So it's a really widespread species. And throughout this large distribution, Peromyscus maniculatus lives across a broad environmental gradient. So this ranges from tropical and temperate rainforests to arid scrublands and deserts. And this species is particularly interesting to biologists because even within a species, there's considerable morphological variation that can be found based on what environment the deer mice are found in. So I'm showing a couple examples of that. On the left here, we have Peromyscus maniculatus from Nebraska sandhills in A, and then the prairie in C. And you can see that they look pretty different. They're the same species, but their pelage really differs based on what environment they're found. And likewise, mice that are found either in forested habitats or open habitats have different number of tail vertebrae, which is pretty interesting. And because of this intraspecific variation, Peromyscus maniculatus has become a model organism for evolutionary and ecological study. So because of their extensive range and this considerable morphological variation, sort of naming the species and subspecies within Peromyscus has been really complex. So there's been at least 60 subspecies of Peromyscus maniculatus that has been described since the original species was described. And then the composition of species and subspecies within the broader Peromyscus maniculatus species group has been modified several times. And researchers have found evidence of cryptic species as molecular techniques have progressed. So I'm just going to talk a little bit about what a cryptic species is, because my talk really focuses a lot on that. So cryptic species are groups of 2 or more taxa that are previously classified as a single species because they look the same. And that's sort of what I'm trying to demonstrate in this figure. But through modern morphometric, molecular, and ecological analyses, hidden diversity within a species has been revealed. So what we once thought was a single mouse because it all looked the same, when we used DNA, it revealed that it was actually several different species or independently evolving lineages. And when we look at the evolutionary history of Peromyscus maniculatus, we can see that the unique genetic lineages sort of correspond to different geographic regions. And this was first noted in Dragou et al., represented on the left, and then several studies have supported these findings, suggesting that distinct lineages occupy a different geographic space. So different Peromyscus maniculatus lineages are happening throughout North America. These studies refrain from proposing taxonomic changes, but they did show evidence of cryptic species within Peromyscus maniculatus itself. So what I'm showing here is the distribution of the species within the Peromyscus maniculatus group. Until 2019, this was the hypothesized distribution of these species. Peromyscus maniculatus, shown in green, having the broadest distribution across most of North America, including large parts of Canada, the United States, and Mexico. But in 2019, this Peromyscus maniculatus species was actually split into 3 different species. So Peromyscus maniculatus now is in the eastern lineage, Peromyscus sonoriensis is in the west, and Peromyscus lobecula is in the south. However, these species were elevated based on mitochondrial DNA only, which does have some limitations. So one of the issues with using mitochondrial DNA is that it's maternally inherited only, which means that the effective population size is a lot lower for mitochondrial genes than it is for nuclear genes. And why that matters is because any genetic differences seen in mitochondrial DNA will be fixed much more quickly than nuclear DNA. So we might just be seeing differences between populations based on dispersal patterns. So if males disperse more often than females in a species, then it'll seem like there's more structure, even if there is sort of gene flow happening between these different geographic populations. Additionally, this molecular loci, the mitochondrial DNA used to elevate Peromyscus subspecies to full species, was unable to resolve the relationships between species in the Peromyscus maniculatus group. So what I'm showing here has a tree that we built from mitochondrial DNA, and the Peromyscus maniculatus group is contained in this box. So what we're seeing is that there's pretty good support for the individual species, but we can't really see support for the relationships between them. So what we're seeing is low support values sort of here and here. So we don't really know what's happening between the species, what their evolutionary relationships are. You know, if these mice look the same and relationships are hard to untangle, like why do we really care if they're separate species or not? But beyond being like just an interesting evolutionary model because they have so much variation, the species is also a reservoir for both established and emerging pathogens that can be transmitted to humans. So these pathogens include hantaviruses, the bacteria that causes Lyme disease, and SARS-CoV-2. So understanding which species is actually harboring these pathogens and where the species exists is actually really important to understand how pathogens might spread. So that brings me really to the fundamental question of all of our research. Where and what is Peromyscus maniculatus? So it's clear that Peromyscus maniculatus, the species, contains multiple clades that might correspond to species. And building off previous research, our goal was to test these species hypotheses using genomic data. If the divergent mtDNA, the mitochondrial lineages identified in countless studies and then named in 2019 actually represent unique evolutionary units, then the identity of species within this important model system might change, which could affect the implication of countless studies. So our specific study goals were to build off these previous studies, to examine the species limits and geographic structure, and using novel genomic data. And what was really important for us is to use museum samples for both our genetic data and for the morphological data that we have. But sort of taking a step back, one thing that really drives our research is a question about what makes a species, like what is good evidence for a species. You know, biologists can maybe broadly agree that they're separately evolving metapopulation lineages, you know, where at one end of this scale, it's very obvious to say there's one species, and at the other end, it's very obvious to say that there are two species. But our research sort of lives in this gray zone where we're unsure if there's one or two species, and we sort of have to look at different lines of evidence to sort that question out. So one way that we could address this is through looking at reproductive isolation. Reproductive isolation is kind of difficult, especially in wild populations, but also fertile hybrids do exist. So that wasn't really, you know, what we were going to do, especially if we wanted to use museum samples. So we are thinking about looking at genetic isolation and morphological divergence. And then we wanted to really understand, you know, the genetic and morphological divergence in the context of different ecology of these different populations, and then, you know, geographic divergence, if there's anything obvious we can tell that are separating these different species. So first I'm going to talk about the genomic side of the project, the part that I really worked on. And our DNA markers of choice for this project were ultra-conserved elements. So we chose ultra-conserved elements because we needed nuclear markers that were powerful enough to detect recent divergence and are repeatable. And we thought about full genomes, but that's not really necessary for the questions that we are trying to answer. We're not really looking for selection or anything like that. We're just really trying to understand sort of the neutral evolution of what's happening in these populations. So we looked for a reduced representation approach. And what we really wanted was something that can cover a lot of different species and is easily repeatable. And there are some genomic methods like RADseq that are really powerful and can provide high resolution data, but they can't really be repeated in different studies. And we really wanted to be able to build upon our research as more and more samples are collected. So we chose ultra conserved elements, sort of a representation of what they might look like is over here on the right. Basically, there's a core region that can be amplified across a lot of different species. And then as we flank out from those core regions, there's a lot of variation in the sequence that will tell us the evolutionary history of the organism. So we used tissues from 15 different museums and a total of 101 specimens. And this is our sampling. So we tried to get a really good representation of the major species, Paramyscus maniculatus, sonoriensis, and lobecula, but also Paramyscus gambili because it seems to have an uncertain evolutionary history with the Western Paramyscus groups. Oh, and there are lots of sampling gaps you see here. That's either areas where there were no samples in museums or when we received the tissue, it was actually the wrong species, which just sort of goes back to what I said about these mice being really hard to tell apart, even by experts. So what we did was first generate phylogenetic trees to untangle the relationships among species in the Paramyscus maniculatus species group. And what I'm showing here are the phylogenetic analyses of the mitogenome. So these data support the species boundaries within Paramyscus maniculatus species group that were proposed in these previous studies, but with higher support for relationships among the clades. So here's the tree. And in this tree, all these nodes with the black dots are full support from both maximum likelihood and Bayesian analyses. And then these numbers represent bootstrap support and posterior probability. And then what you're looking at here is the map representing the sampled individuals colored with the clade identity. So the clade over here corresponds with these colors over here. So these analyses support the general species boundaries, and they strongly support Paramyscus polienotis, which is found in Florida, a sister to the remaining species group of Paramyscus maniculatus. And importantly, the recently elevated subspecies are all sister to each other, and then the Western species are sister to each other in a different clade. And why I'm pointing this out is because when we looked at the nuclear data, we found different topology in our tree. Most notably, Paramyscus maniculatus and Paramyscus polienotis are actually sister species in this tree. So instead of the newly elevated subspecies being, sorry, I'm looking at questions. I'll answer all your questions later, sorry. But the triangles basically just represent there's many individuals happening in each of these clades. So, there's maybe like 20 of each sample of the species. So instead of these newly elevated species being sister species, they're actually more separated by geography. And really importantly, Paramyscus sonoriensis is not monophyletic, as it was in the mitochondria analyses. And we can see that they're also not geographically separated. These purple and yellow clades intermix throughout the species range. And we also conducted species delimitation analyses. So these are a little different than tree-based analyses. Rather than trying to look at the relationship among the species, we're just trying to understand the support for each clade, the support for each individual species. And the method we chose was Bayesian phylogenetics and phylogeography. So basically, we're looking at, we used only 500 of our loci to test the species, but they did have high support. So each species had a lot of support for it being a species and not just a different lineage of the same species. So these mitogenomic analyses did support the recent findings of species limits within Peromyscus maniculatus group. However, the UCE trees have different topology. And then, like I said, the species delimitation support the name species group, the name species within the Peromyscus maniculatus group. And so our next steps are to understand why we see differences in the tree topology. Could this potentially be due to that differences in effective population size that I mentioned earlier between mitochondrial and nuclear loci? It's possible that there has been insufficient time to allow nuclear loci to reflect the isolation seen in mitochondrial analyses, or there's potentially ongoing gene flow between these different subspecies or species, and then there could be historical introgression or hybridization between the species. So we're trying to look at different methods to sort of untangle why we're seeing differences in our mitochondrial and nuclear trees. And that sort of brings me to talking about morphological variation in deer mice. So although, these mice might seem superficially the same, there is potential for variation that corresponds to species boundaries or ecological differences within species. So our main question is really to understand, are there morphological differences that track with the genetic data? So we have these cryptic species that clearly show strong genetic differences, and we wanted to explore the morphological differences. So the graduate student in our lab, Grace, is looking at external differences in the different species of Peromyscus. So she's using external linear measurements, so that's just the length of the body, the tail, the hind foot, and ear lengths, because those can be able to tell Peromyscus species apart. However, what she found sort of looking at these charts is that with the exception of Peromyscus polianotus, there's not a lot of differences that we can see in sort of body length or tail length that really separates these species. So what she's interested in now is exploring if these traits are not following species boundaries. Are they perhaps a product of convergent evolution based on ecological characteristics? So are species that are living in similar habitats having similar response to environmental pressures such as temperature, precipitation amounts, their diet, having to go through the same habitat. So she's sort of exploring different ways to sort of parse the differences between Peromyscus based on their external characteristics. And then 2 graduate students in the group are looking at 3D morphometrics, understanding if it's maybe shape rather than size that is a useful tool for exploring differences among Peromyscus. This is really cool because CT scanning is non-destructive. So there are some specimens that are skeletal and some that are fluid preserved, and we can take advantage of all of these types of specimens to look at internal and external characteristics of Peromyscus. And then we can also upload our CT scans to MorphoSource, which is an open access sort of data repository that will allow people to build off of our work, repeat our work, or have new hypotheses. So it's really exciting. CT scanning is pretty cool. And this is just an example of what one of our CT scan Peromyscus would look like. So you can see that we can look at both the size and the shape of the Peromyscus using these scans. And the goals of the 3D morphological studies were to look at different ways of exploring cryptic species. So do morphological differences correspond to habitat differences rather than species differences? So to explore this question, the graduate students, Brandy and Haley, are looking at differences between mice that are found in forested habitats and mice that are found in open or prairie habitats. Why they think there might be a difference is, you might have guessed this, but mice in forested habitats are more likely to be arboreal compared to mice that are found in open environments. And they think that, maybe there's some sort of difference between these mice. And there has been recorded differences in tail length between specimens found in open habitats versus specimens found in the forest. So, we just wanted to look at different skeletal structures to see if there's any other variation. So what Brandy is looking at is cranial morphology. She's looking at the size and shape of the skull and the mandible. So those are the skull and the mandible. So they're developmentally independent yet functionally integrated. So, the mandible might show differences and the skull might not, or vice versa. So she's looking at both structures. And why she's doing this is that past studies have shown that the shape changes related to muscle insertion. Figure this out again. Okay. Share my screen, start the presentation. Great. Can you guys see my presentation?
Looks good. Thank you.
Okay, perfect, cool. Sorry about that. Fantastic. Okay, so Brandy, like I said, looking at skull morphology, and what she found looking at the cranium is that her preliminary analyses make it seem like there is a difference between the forested and non-forested individuals. So looking at this PCA, we see that, there's maybe some structure, some clustering of individuals based upon where they're found. Since this was a preliminary study, the p-value isn't technically significant, but she is adding more specimens. And as she does, this value could change. However, the preliminary mandibular measurements don't really show any differences between the forested and non-forested habitat, which is really interesting because, you would expect that if it's dietary preferences, that there would be changes to the mandible. So it could be something else that's driving differences in skull shape rather than diet, which is pretty cool. And then Haley is looking at limb morphology. So she's using landmarks to look at both the shape and then linear measurements to look at the size. And this is because limbs are linked to locomotion style. So you can imagine that semi-aquatic rodents might have more robust bones and large muscular attachments, short femurs, elongated hind feet. Arboreal rodents, longer humerus, longer digits, equally proportioned hind and forelimbs. Rodents that are fossorial, they'll be this presentation. There's a chance that'll happen again. I don't know.
If
We
you
do
can.
third time's the charm.
Yeah, maybe.
Give it one
Okay.
more shot.
Thanks for bearing with me.
Sorry you have to go through that. I can understand how frustrating that must be.
Yeah, it works every day up until I need it to work. So we'll do what we can. Okay. Cool. Hind limbs. So she was looking at the femur, tibia, and the fibula. That's what she's looking at in the hind limbs. And what she found really when looking at shape is that the only real difference was in the shape of the femur. So we're seeing like a pretty good evidence of why femur shape might be different, that femur shape might be different. And then as she goes along, Haley's going to explore, why there might be differences in this femur shape. Is it longer? Is it shorter? Why might that be? So she's sort of excited about this, but she's still working through it. And then she also found differences in the length of these structures when looking at animals that are in forested versus open habitats. So we've got the radius and the femur. So looking at this, there might be arboreal species. The individuals that are in forested habitats might be, more adept to climbing, even if they're the same species as these open habitats. So what she's seeing is that sort of these morphological differences aren't partitioning along species lines, sort of in line with what Grace and Brandy found. So, what's the big picture? What have we found out about Peromyscus in North America? So the mitogenomic data found strong support for the named species and relationships, but the nuclear data is less clear. So there's different topology between our mitogenomic and nuclear data, and we're still exploring, like I said, possible integration, gene flow, or hybridization that may be underlying this mitonuclear discordance. Because we did find that the high support for each species using the nuclear loci. So we're just trying to understand, what is happening with this discordance in the evolutionary relationships. And there is evidence that morphological variation is stronger between the ecological gradients rather than along species lines. But we do need to sort of untangle the morphological variation found within species. This is kind of difficult because, when we were sort of planning this study, we requested stuff from museums, anticipating that each specimen was going to be ID'd to species correctly. But that's not always the case, especially when the Paramiscus maniculatus is in sympatry, living in the same place with Paramiscus leucopus. A different study showed that up to 30% of museum specimens were mis-ID'd to species level. So we're trying to untangle how we can, reconcile our CT scan data that doesn't have genetic data attached to it to make sure that we really have the right species. So that was pretty interesting, and sort of also brings me to my next point about, the importance of museum collections and why we, hold on to stuff from previous studies, without sort of holding on to genetic data from all these previous studies, we wouldn't have been able to understand that these species were misidentified. So yeah, natural history museums are more than exhibits. And, one goal of this project was really to use existing museum resources. And I just want to talk about some cool things that I think that museums do. So, natural history museums, many of them have public exhibits that serve as a way for scientists to share research and discoveries with the public. And I think that this is really important. One of the reasons why I even became a scientist is because I got to go to a natural history museum as a kid. So I think that this public part of museums is really important. But there is also a large scientific collection that's used for research in museums. Not all of them have public facing fronts, maybe just open houses where you can see, the depths of the dungeons, as I like to call it, where there's no light to keep our museum specimens preserved. But, there's a lot happening beneath the public sphere. And this is important because specimens sort of act like time machines. They can document environmental change over centuries. And, one really interesting study found coloration in birds in museum collections. So these birds were used to track the amount of black carbon in the air over time, and, can track the effects of environmental policy upon pollution. And as ecosystems are modified, sometimes destroyed, biological collections become, the only remaining representatives of endangered species. And they can also help document the decline of species as habitats become fragmented. So, we can have records of a species that was found, at a high elevation or a low elevation, in 1850 and 1900, but now our new collections are showing that there's much less of this animal being collected at these elevations. So it can really track different distributions for species. And then something that I've been finding recently really cool is how researchers can use museum collections to sort of track the history of infectious diseases. Connecting the site of a disease outbreak to a potential pathogen could help and host that, you can pinpoint the area and can help pinpoint the sources and pathways of transmission. And what's really cool to me is, preserved specimens such as rodents can carry these preserved pathogens, such as pox viruses or hantaviruses. And this research could really help build a faster response to emerging pandemics, and we can better predict where and how diseases might appear if we know which animals they, have historically been found in. There's a lot of specimens in museums already. Why would we want to keep adding more? Researchers do actively collect new specimens. Ecosystems and species distributions, like I said, are always changing. And these new specimens can help document current conditions so we can track changes over time. And without these continuously updated collections, we have an incomplete and outdated view of biodiversity. And something that I think is really cool is genomic resource centers. This is the one that's at the University of Alaska Museum. And this really helps long-term preservation of tissues that can aid in countless studies, evolutionary studies, um, conservation studies. So I think that as we have updated preservation techniques, collecting more data, more specimens is going to be really important. And along with that, the extended specimen is something that is talked a lot about in museum research now. So this is, more detailed metadata. So where the animal was collected, what the weather was like, how many animals were found in that area are, that data is associated with a physical specimen and uploaded, to the internet and the sort of public databases. And that's really cool because, you can use these databases to access information, conduct studies within your classrooms because, you have access to these CT scans, these measurements, genetic data from specimens. So it's really cool, you can really build new and interesting studies based on data that already exists. And I think that researchers do a lot of things to support collections, but just, talking to museum curators in your area so you can just deposit your specimens once you're done and then provide all this metadata for digitization. And then, museums are like, I don't know, animal libraries. They, curators love it to loan or to visit. So we really, we love it when people use museum specimens in new and interesting ways. And it also sort of cuts down on the cost of your studies too, if you don't have to go out and collect new data. And then when you publish studies using specimens, just acknowledge the collections sort of use publications based on their collections, sort of in grant proposals and stuff, just to show how important the collections are. And so that's sort of all. Just, I think that, this is one study that I've done with museums. I've done several other studies. I currently work in a museum. I did at Texas A&M and at Ohio State. So yeah. But I do want to thank Ira for sharing his insight. He has extensive knowledge of Paramescus. Emma Lemberg, trying to get DNA out of historic specimens is not as hard as it used to be, but we did have some trial and error, and then just all the Light Lab members for their helpful feedback. Okay, and despite the internet being bad, I can take any questions now.
Thanks, Natalie, and thanks for rolling with that. I know how disheartening that can be and frustrating. I'm trying to flow through your materials and technology gets in the way.
Yeah.
But if folks have any questions, I know we had a couple previously. I think we lost at least one of the people asking the question.
Okay.
Yeah, feel free to unmute or put it in the chat. I wanted to echo some of this that I actually had forgot about this till you mentioned it. That I was fortunate enough to see some of Darwin's original collections in the British Museum of Natural History in London, and that was just like a step back in time. It was humbling to be in the presence of such an amazing naturalist and scientist. But I think there's so many applications for these samples potentially, maybe for finishing or adding to stable isotope libraries, for example, for diet analysis. That's actually one of the reasons I reached out to Link in the first place. Yeah, so many applications. Yeah, I know there's a question about were you able to derive complete genotypes, mitochondrial genotypes from your museum samples, which I assume is implied, but
Yes, we were. So,
about the quality of those or any challenges you had, perhaps?
yeah, sorry, when my internet gave out, I can't see the chat questions anymore. So, yeah, we were able to get full mitochondrial genomes for, I think, 90, which is about 90% of our samples. So some of them with the lower quality data, we couldn't get the mitogenomes, but it was, they were complete genomes, mitogenomes. So that's pretty cool. One of the sort of benefits of using UCEs.
That's pretty amazing. It's come a long way.
Yeah.
And I alluded this during when you were gone, and then we talked about it briefly before your presentation, but do you think there's potential for looking at, say, DNA methylation, epigenetic studies using museum samples, or would they tend to be too degraded, perhaps?
I think that the methods of whole genome resequencing are getting pretty good at using degraded DNA. I don't really know a lot about methylation, but you can do, sort of selective studies looking at, genome-wide associations from museum samples, and you can get pretty good whole genomes. So I think that there's a lot of applications, especially as genomic technology gets more advanced.
Natalie, I had a question about the slide you were showing where there was a phylogenetic tree that you constructed using UCEs, and there were colored triangles at the as the leaves of the trees, and they had different sizes and dimensions. I was wondering if this is probably me not knowing enough about how those trees are constructed, but um, do the sizes of the triangles indicate anything or communicate anything in particular? I noticed that they were all uniquely shaped.
Yeah, the different sizes do correspond with sort of how many samples have been collapsed into that clade. So basically, showing a simplified version where, everything that is Paramescus maniculatus gets lumped down into one sphere or triangle, and we had different numbers of samples for each of those clades. So yeah, the triangles are just based on sample size.
Gotcha.
Yeah.
Thank you for clarifying.
Mhm.
I just wanted to share like a personal note on you talking about your inspiration from going to a museum and that inspiring you to become a naturalist. Um, I also saw a similar talk by um, the head of, I believe it's data science and information technology at the Zoological Society of London. They were using a collection of GBIF data to create the planetary knowledge base. And I was just trying to look up if they actually have this, but I believe they actually are using like their entire archive of samples there, and also a network of museums across the globe, including the American Natural History Museum in New York, who kind of tag and digitize all of the species that they've archived over time, and also tag them with their whole genome sequences. And I think they have like a time series of those as well. And that was super
Yeah,
inspiring. Like I'd never heard of that before, so it really opened up my eyes to the value of natural history collections like those.
yeah, it's pretty cool. There's I feel like especially undergraduates working at museums, there's lots of digitizing to do. So yeah, it's really useful stuff when you're like, I wonder, is there this species, with measurements or like a picture that I can take measurements from, and you know, more and more there are ways to like look that up remotely, so you don't even have to go to the museum or request specimens. So it's pretty cool. And then yeah, they can associate them with their like GenBank accession. So anything that you do with genetically can be associated with the physical specimen. Yeah. So yeah, it's pretty cool.
That was another thing that caught my attention was the interface, the georeference database that the Museum of the North has. It was just blew my mind, the amount of data and access to samples, ability to query samples, see their history. Yeah, it was incredible to see.
Yeah, like a really cool forward-looking application that I heard about in that planetary knowledge base talk was the way they have it set up, they can actually run machine learning inferences on those data sets to predict what species may not have been discovered yet to drive further discovery studies, which was really, really cool.
That is cool. That is really cool.
Great. Well, I haven't said this in the past, but if folks want to try and connect and follow up on any of these talks or share resources, like links to maybe the publications, right now we don't have a great system for that, but through LinkedIn or some of the other social media type sites, I think people would appreciate hearing some of these stories or seeing more about them. Any other questions for Natalie or myself or anybody else? Of course, we're both available through email. Always welcome questions. It's nice to hear from other folks for sure. And I do want to thank everyone again for sticking through. I know, again, frustrating losing power. I'm glad it was you and not me. I have to say that.